Thank you, Alice, for inviting me to this webinar. So I'm Marc Dumas. I'm Chair in Systems Medicine at Imperial College, and I'm also Director of Research at CNRS in Lille. And I'm also a visiting professor, an adjunct professor at McGill University. So when we were discussing about this webinar, Alice asked me to talk about something different from the podcast we have recorded. So last time for the podcast, we talked about fatty liver disease, and we already had a speaker about fatty liver disease. So this time I'm going to talk about more how we use the metabolome and microbiome data, how we leverage the data in epidemiological or clinical studies. But this time as a little more in cardiometabolic disease contexts. So I think we've already had a good start at this. So one could easily challenge the accepted view that we are a product of our genes. Well, then maybe we should just reframe that question saying, well, actually, which set of genes are we talking about? So by the current estimates, we have at least 1,000 times more microbial genes that have been cataloged than human genes. So that means, it doesn't mean that every single one of us has 20 million genes. It's less than that, but it is a versatile constellation of genes and organisms, and we are we are a mosaic of those organisms, which complement our own genome, and that's actually an awful lot of it's a reservoir of functionality, which has been relatively untapped until now, and as a biochemist or as a failed biochemist, I would probably tend to argue that it's not necessarily just the bugs names that are important. It's also what they do. It's also the metabolites that they produce, and actually, are they biologically active? So, a lot of notions I come across a lot of notions. I thought I should probably very quickly whiz through a few of these notions in order to lay the groundwork for the talk, for the introduction of the talk. And well, of course, we meet our microbiomes really early on in life as we get born. So usually when it really goes through the birth canal, for the first few years, we have a very homogeneous nutrition, which is made of a very challenging matrix, analytically speaking. I'm talking about milk. But I think we all know what it looks like, what it tastes like. And the bugs in our guts really love it. And there was actually recently, as of this week, a very interesting paper in Nature Communications that's complementing what's on this screen. And there's an explosion of microorganisms when we move to solid foods. And for those of us who have been changing nappies, then you can see a very different change, a very different set of feces that are being produced as you introduce solid foods into the diet of the infants and the toddlers. And after that, it's just like stamp collecting. It's very difficult once you have made friends with the bacteria. It's actually very difficult to get rid of them. And it's only much later on in life, with frailty, that we see much later on a decrease in this diversity of bacteria, viruses, and so on, and fungi, and the genetic diversity of those microorganisms. There were a few concepts, and some of them were transformational, even though maybe some of them were transitory. And one of them was this notion that there is this ratio between bacteroidetes and firmicutes. It was introduced in 2 papers back to back just before Christmas in 2006 by Jeff Gordon's group. And you can recognize names of leading figures in the field who were postdocs or senior postdocs at the time in Jeff Gordon's group, and this notion that you had two polar opposites, the firmicutes and the bacteroidetes phyla. So it's quite high up in terms of taxonomy. So very, very different types of organisms which were dominating the ecologies of volunteers with or without obesity. And this was shown to be in the causal relationship through fecal microbiota transference in mice, both using microbiota from genetically altered mice, but also microbiota from donors. So this notion that you are somewhere on this continuum, and maybe you're more on that side, or maybe you're more on the other. Very quickly, After that, our friends 5 years later, actually 3 years later, our friends from the MetaHit Consortium came up with this idea and those results that actually maybe it's not just 2 points on the line, maybe it's 3 points, maybe it's a hyperplane where we have this clustering of bacterial communities into what we called, what was called at the time, enterotypes. So types of ecological communities in the gut. And it's not 2, it's 3 types of communities. So all of a sudden, it's opening up the field. It's not monodimensional. It's at least bidimensional, and we have a plane to look at. The name enterotype was, it was modeled after the blood types, but I think the blood types were a bit too discrete. What they are describing here and what they were describing here is, and you can see very different types of communities, and it looks like that they are discrete communities here, but in fact, the notion it's a bit more like you're looking at a map, a 3D map. So if you're in Austria, you can see the Alps, you can see the mountains behind the windows and in the background, and it really depends where you put the contour line, and then you can end up with 2 or 3 or maybe more, maybe 4 enterotypes in your communities. And now there's even more work that has been published where there are in fact consensus data emerging saying that it would be more likely to be 3 enterotypes across the world. But some of these enterotypes only show up in situations of, but this is population data. There is some evidence that maybe there's some enterotypes that show up in situations of disease, such as metabolic disease or such as poor gut health. If we go back to the metabolomics, then I wouldn't be doing a good job if I wasn't telling you that, okay, we have this collapse of microbial gene richness in obesity. So pretty much, if you start from a 10 million gene atlas, you end up having about, 700 to 800,000 genes in our guts. So these were the original results. This is what we see here on the graph here. And then in situations of obesity, there's a shift of that distribution towards the left. And then you end up with, you're losing about 40% of this diversity in situation of obesity. So clearly we know about the burden of the metabolic diseases. We know about the determinants. A lot of my esteemed colleagues, I spend a lot of time looking into the genetics, into the lifestyle. One of the keywords that's popping up more and more and more, and then we should probably think about it, is the exposome. But for now, looking at the microbiome, we have seen that there is this collapse in diversity from the microbiome. So presumably, what these microbiomes do, we're probably losing some of the good guys, and we're probably gaining some of the bad guys if we want to make it very simplistic. And probably if we frame it in terms of molecule, we've been losing some of the good molecules, maybe the probiotics, the probiotics and the postbiotics, what the bugs make. And we've been gaining some of the bad guys, some of the bad molecules, maybe the pro-inflammatory compounds. We were talking about LPS earlier, lipopolysaccharides, and also peptidoglycans. I mean, these are relatively abrasive metabolites. They're going to trigger a pro-inflammatory response. Most of the metabolites made by the gut microbiome, usually when we look at the small molecules, most of them, the vast majority of them, are actually beneficial compounds that seem to tone down the immune system and improve metabolic homeostasis. So how did we get into this? Well, that was a long time ago, by chance. So we talk a lot about serendipity, but by chance we were looking at a signature of fatty liver disease and insulin resistance. And then we found that everything was evolving around the metabolism of choline and the production of microbial metabolites in situation of insulin resistance. And you have 2 strains of mice, some on low choline and others on the high choline diet. Low fat and high fat diet. And then we found that that was evolving around the metabolism of choline, and the mice on a high fat diet were getting steatosis. And one of the key hallmarks of this high fat diet is that the mice were excreting an awful lot of TMA, TMA1, DMA. But what went flying a little bit below the radar is that only the 129 strain, which gets fatty liver disease, was showing a low levels of fat, or at least low bioavailability of phosphatidylcholine in the plasma. Sorry. So the thinking at the time is that, if you modulate choline, choline deficient diets, they give you fatty liver disease. So we thought that because the bugs chew choline in the gut, choline is not bioavailable. And if you don't have choline, you can't make the LDL. And if you can't make the LDL, you can't export fat from the liver. So that was the thinking at the time. In the meantime, so it was in 2006, a few years after, Stan Hazen took the field by storm on the cardiovascular side of things with a first paper and then a series of other papers that basically hammered the message that TMAO is bad news for you in terms of cardiovascular disease, and with a lot of evidence basically showing that in that case TMAO might not be such a great idea. And it's more about atherosclerosis more than atherosclerosis by itself. But it looks like TMAO is triggering platelet aggregation, which can then generate long-term adverse outcomes when it comes to cardiovascular diseases. What we looked into, so fast forward a few years down the line, what we published a couple of years ago with a very good research fellow in the lab, Petros Andrikopoulos, is that we looked at TMAO, but under a different light. We looked at kidney function and kidney fibrosis, and when Petros came to the lab, he really wanted to look at TMAO under this kidney angle, and we looked into some of the data sets we had in the lab. So one of them is based on the metacardy study, and we built a machine learning model, using gradient boosting and SHAP to rank basically the variables. And what Petros showed is that out of all the contributors and all the determinants that we had when we put all the geomics data and clinical data together, well, number one for TMAO concentration this age, I'm afraid there's nothing I can do about age. And number two was kidney function, EGFR. And that there's a lot you can do if you look after your kidneys properly. And what Petros did as well in mouse models, based on these results, is that he looked into how TMAO and choline administered to mice when you make a ligature of one kidney and you leave the other kidney unaffected. So you have, in terms of statistics, green because you have paired statistics. So you got two kidneys, one is the control and the other one is the affected one. And you can see that when you add TMAO, then you have an increase in alpha SMA and markers of fibrosis in the kidneys of these mice. And maybe the silver lining and the take-home message and the interesting bit in terms of translational medicine is that when you focus on the renoprotective medication, anti-diabetic medication, so GLP-1 receptor agonists, so in the metacardy study, so the patients who were prescribed GLP-1 receptor agonist, they tended to have less TMAO in their blood. So this is an association. This is not clinical trial. So we don't have a before and after, but it seems that treatment with renoprotective anti-diabetic medication seems to protect the kidneys, and as a consequence, you have an improvement in, or at least you maintain the capacity to filter, to excrete TMAO into urine, and therefore you have some added benefits in terms of cardiovascular risk. Yes, so this is a visual. This is a graphical abstract, basically just summarizing what I just said about this study and some predicted. Maybe that would be one of the possibilities for cardioprotection later on the line. Another metabolite that's really interesting is hippurate. So hippurate is a phenylpropanoid metabolite. So it's at the bottom of the phenylpropanoid pathway. Here, that was in another study that we performed in metacardy in the MetaHit dataset. And you can see that all these metabolites are in the phenylpropanoid pathway. We only showed the ones that are significantly associated with hippurate. But you can see that this is all funneling towards the formation of benzoic acid, which then gets uptaken and gets conjugated in the liver with glycine. So this is phase 2 drug metabolism applied to natural products here. And you can see that not only we can predict gene richness using metabolomics, so this was a urine NMR at the time, and then one of the metabolites the most significantly affected with gene richness in urine was actually hippuric acid, hippurate. And so hippurate, if high gene richness is good for you in terms of metabolic homeostasis, and therefore hippurate must be good news as well. What we've shown is that people who were who were tending to take medication on a high-fat diet tended to be more tended to be more responsive. What people who had high hippurate, when they were taking a high-fat diet, tended to control their glycemia much better and their insulin resistance much better. And in fact, it seems that hippurate contributes to a lot of the association between gene richness, gene count, and OMA, insulin resistance. So you can, that's not exactly a mediation analysis. We should have done a mediation analysis, but we didn't at the time. But this partial correlation analysis, which is very similar, is kind of suggesting that we would have a mediating effect of hippurate when it comes to the association between gene richness and insulin sensitivity. More recently, this is just off the press. We just got the paper accepted and published online over the Christmas break. We've looked into a lot of the work that has been done on hippurate by ourselves and by others has been done mostly in biofluids, so mostly plasma, but also urine. And here, thanks to François Patou in Lille and his ABOS study, where we were fortunate enough to look into plasma and liver levels of hippurate, and not only you can see that the plasma levels are it's a significant correlation, but you can see there's quite a lot of spread. It is kind of a good estimate or a good proxy of liver hippurate. And when you look at how liver and plasma hippurate associate with the different components of the NAFLD score, you can see that it is negatively associated with steatosis, with inflammation, with ballooning, with fibrosis, and with the NAS score itself. So it looks like it tends to say that, at least in terms of association, hippurate tends to be good news when it comes to fatty liver disease. We looked into the gene expression, hepatic gene expression, and what we found is that hippurate tends, again, it shouldn't be a surprise, to negatively associate with expressions of genes involved in measles and MASH in the ABOS population, which triggered us into testing the role of hippurate on human hepatocytes. And as you can see, addition of hippurate in situation of chronic insulin treatment, which is triggering at least a form of insulin resistance in cellular models, you can see that when you add insulin, you increase chronic insulin for over 24 hours, you increase lipid storage in the cells, and then when you add hippurate, you kind of normalize this, and then you see also something that's reminiscent of restoring insulin sensitivity in those cells when you then perform some glucose uptake assays. For the next part of the talk, and I'm going to wrap up relatively quickly because otherwise we're going to run out of time. For the next part of the talk, what I'd like to get the audience, to get you to think about is the importance of drugs and medication in what we do. Most of the samples we're looking at, the vast majority of the human samples we're looking at, these are not, I would say, clinical studies. These are not clinical trials, so we're not completely controlling everything. And what we have is a collection of patients, and because we didn't necessarily have patients at diagnosis, so a lot of them are already medicated. They are seeing their doctors and then their specialists, and they are being prescribed drugs. And what we saw with Per Bork's team at Heidelberg and Sophia Forschland, who was working for Per Bork at the time, is that in 2,000 patients we had about 1,000 different molecules, and you can see the effect of polypharmacy prescription of medication. So some patients had at least 8 molecules being prescribed. And you can see the association clusters of the different molecules. We could spend a lot of time looking at those, but you basically have a cluster on the cardio side, and then you have more a cluster on the metabolic side of things that you can see here. I mean, the patients were recruited quite a long time ago, so it was 12 to 15 years ago. So that explains also some of the patterns of the medication that they're given. What I'd like you to think about is that those medications that our patients are taking, and that of course, with metabolomics, not only we have the perfect readout to see some of these compounds, this exposure, but we also have some really good goggles to look at the impact of medication on the metabolome, on the disease itself. So part of this study, and what Sophia and the team did, they looked into the associations between the types of drugs and the different markers we had. So we had a lot of microbiome features here, and then we had a lot of host markers. For some various reasons, the metabolomics ended up with the host features because we were looking at plasma and urine, but essentially a lot of microbiome metabolites popped up there. And it shouldn't be a surprise, but the biggest association was, in fact, acetylsalicylic acid seen by GCMS being associated with aspirin prescription. So we have essentially a direct analyte to estimate the direct exposure, or at least the bioavailability of the drug in that case. What we then did next, and that's Kantachichi, who was and is a research fellow in the lab, we looked into the deconfounded signatures of the metabolome and the microbiome, and Sophia Forslund came with this algorithm for deconfounding the impact of drugs. And as you can see, it took us believe it or not, but it took us a long time to come to that simplicity in the visuals, in a visual simplicity that you have when you move from metabolic health to metabolic disease, and from metabolic disease to cardiovascular disease. So that's kind of recapitulating the natural history of the cardiometabolic cluster. So you get from healthy to metabolic disease, and then like in your 30s and 40s, and then in your 50s and 60s, this is when you start developing cardiovascular diseases. And when we look at the associations, it took us a long while to come up with something that simple in terms of trajectories, but we found markers that are associated with cardiovascular disease, ischemic heart disease, which we used to call coronary artery disease before. So there are markers only associated with cardiovascular, with AHD, but not associated with metabolic disease. Vice versa, there are markers of metabolic disease that don't show any further change when you go on to develop cardiovascular disease. Some markers are escalating, so you can see the accumulating burden of those markers. And quite interestingly, some markers seem to go up in metabolic disease and they go back down in cardiovascular disease. And for that, there's a lot of possibilities. One is the fact that you don't have patients that are drug naive. So maybe the drugs they are taking is actually working and it's improving their metabolome and their microbiome. The other ones that they're also acutely aware of their health condition. So it usually tends to result in people taking some proactive steps in terms of personal lifestyle, so more exercise and a healthier diet. So you could see normalization of those. And there's also survivor bias. So the people whose metabolic profile was so adverse are no longer here to talk about it because they've probably already experienced a massive cardiovascular event. I mean, I didn't make it. Interestingly, when we go and look at the escalating features, you can see here the features escalating from health to metabolic disease and from metabolic disease to cardiovascular disease. When you look at the colors of the markers, the vast majority of the markers, the names, the labels, you can see the red, that means metabolites, and we only had one taxon, Rhinococcus, which was actually directly obtained from the microbiome. And here again, when you look at the collection of markers, even though we have lots of bugs and lots of genes, The good news for people who do metabolomics is that the metabolomics variables tend to be a lot sharper than the metagenomics data. And the metagenomics data are beautiful, but it looks like the metabolomics data really capture some of these trajectories. When we came up with a metabolomic and metabolome and microbiome machine learning model to predict IHD. So these are the calibration data. So you shouldn't really looking at the ROC curve because it's the calibration data. So of course, everything's perfect in an ideal world. What you should be looking at is the real performance when we use an external data set. So we trained the model on the left and we applied the model on external data on the right from our friends and colleagues, Eran Segal in Israel. And you can see that the performance of the clinical risk factors, Framingham risk scores and so on, was good, but it wasn't perfect. When we added not when we added the metabolomic and microbiome data only in the model 2 predict much better. And then when we merge the two, the clinical and the omics data, then we have an improvement in the performance of the ROC curve here. And interestingly, I just mentioned that we exchanged data with Eran Segal, and in their paper, they were looking at various metabolites and bugs. And again, there's a lot of metabolites that are very close to my heart, at least my scientific heart, my scientific interests. And here in that case, they were looking only at acute coronary syndrome. So people having essentially just having had a heart attack. And then you can see again that there's lots of macrometabolites here that are really predictive and being strongly associated with SES, but also metabolic homeostasis, the contribution of the microbiome, diet, traditional risk factors, and the genetics here. So at this stage, I should probably wind up very quickly. Thank all my collaborators, the funders, all the people I've had the pleasure to work with. Obviously, I'm just the guy who's talking to you about it. And I guess this is all for now. And I'll thank you for your attention. And I guess we'll back to the questions.